Welcome to IGCSE Survival Kit by AZ. Subscribe now and survive IGCSE. Hey, welcome back IGCSE warriors. Today we will be revising the Biology 0610 syllabus, okay, and we will do it as quick as possible in the fastest way. So it will be the full syllabus revision, and come along with me. Don't forget to subscribe to this channel. And more aware, I think most of you all must be having your paper 2 exam tomorrow. All the best for that. So let's revise the syllabus together. Okay, so starting with chapter 1, I'm going to be really fast, okay? Oh my god. Okay, starting with topic 1. Okay, the characteristics. Okay, so topic 1, characteristics and classification of living organisms. So basically, how do you describe the characteristic? How do you tell if an organism is living? These are the characteristics. And you will remember this using Mrs. Gren. Okay? Mrs. Gren. M stands for movement, R stands for respiration, sensitivity, growth, reproduction, excretion, nutrition. Okay, now let's look at the definition. Movement is an action by an organism or part of an organism that causes a change of position or place. Okay, the change of position of the object changes, the organism changes. Respiration as a chemical reaction in cells that break down nutrient molecule and release energy for metabolism. Glucose, basically glucose plus oxygen gives carbon dioxide plus water. This is the reaction we are talking about over here, and energy is also released. Okay, and you get the energy for metabolism. By what is metabolism? Metabolism is all the chemical reactions that are taking place in the cell. Now, sensitivity, it is the ability to detect or respond to changes in the internal or external environment. Any change, internal environment, it can be blood temperature, external environment, it can be if you detect you can see maybe dust you detect, so your eyes suddenly close, you detect any reflex action, someone comes and does something near you, so your eye closes. That is sensitivity. Next, for growth, it's a permanent increase in size and dry mass. Your size and dry mass permanently increases, means it is permanent, it can never come back to its original position. One mistake students do is that they think that the puffer fish, I think that's what we call it, yeah, when it swallows water it grows bigger, so yeah, so it has grew, but that is false. But because it comes under sensitivity, and when it swallows water and then after that it comes back to its original shape. I hope you got my point. Reproduction, as the processes that make more of the same kind of organism, same kind of organism is produced, and it's same, okay, and more of it is produced. Excretion as the removal of waste products of metabolism, like carbon dioxide. After respiration takes place, carbon dioxide is removed from the body, and substances in excess of requirement. Any substances that are in excess of requirement, like we remove amino acids from our body in urine. Okay, but okay, next, nutrition, as the taking enough nutrients for energy, growth, and development. Make sure you know all these definitions by heart, especially for paper 4. Next, state that organisms can be classified into groups based on the features they share. Like if they have, if they all have a backbone, they'll be under vertebrates. If they all don't have a backbone, they'll be under invertebrates. Okay, then species, as a group of organisms that can reproduce to produce fertile offspring. Okay. Okay, binomial system is a system of naming species as an internationally agreed system in which the scientific name is made up of 2 parts showing the genus and the species. For us, it is Homo sapiens. Okay, so Homo sapiens. Here the genus will be capital, the species, the letter will be small, the first letter. All right, Homo is Homo. This is the genus. Okay, this is the genus. Oh, I'm so sorry. Genus. Okay, and this is the species. Got my point? Okay, so next, sequences of bases in DNA are used as a means of classification. Other means of classification can also be used, like morphology, anatomy. Okay, anatomy is looking inside at their bones. Morphology is looking at their physical features. Alright, explain that groups of organisms which share more common ancestor, more recent ancestor, are more closely related and have base sequences DNA that are more similar to that are distant. There's nothing to explain this. It's pretty simple. Make sure you know how to use dichotomous keys. Practice questions on that. Next, there are main features to place animal and plants into the appropriate kingdoms. I'll tell you all very quickly. For animals, they must have a nucleus. They must have all have a cytoplasm. Okay, they have no cell wall. They have no chloroplast. They feed on organic substances made by other living organisms. For plants, okay, they can make their own food by photosynthesis. They have chloroplasts. Okay, and what else? In plants, basically they also have a nucleus. They have cell wall. Remember, and there is cell wall. Okay, they have cell wall, and their cell wall is made up of cellulose. Okay, cellulose. and plant cells do plants and animal cells, sorry, they don't have a cell wall. Okay, and remember that not all plant cells have a chloroplast. There was a question asked about what if all plant cells have, okay, and all was, I believe, highlighted, and there were options like cell wall, chloroplast and all. So not all plant cells have chloroplast, okay, remember that, but most of them do. All right, then main features place into all of the 5 kingdoms. Okay, there are 5 kingdoms: animal, plants, fungus, prokaryote, protoptist. Let's look at them in detail. Animal we have already discussed, but under animal, okay, under animal there are 2 more kingdoms. There are 2 more classifications. One is invertebrate, okay, one is invertebrate and one is vertebrate. Looking at invertebrates, first of all, they're invertebrates. They're also called, there are many types of invertebrates, but we will be dealing with arthropods. Okay, so let's have a closer look at arthropods. Okay, we will do it here. Okay, so arthropods, basically arthropods. Okay, so basically first we will be dealing with insects. Remember, insects, they have, I will be telling you very quickly. Insects, they have 3 pairs of jointed legs. 3 pairs of legs. Okay, then they have 2 pairs of wings. 2 pair of wings. All right, then they have 1 pair of antennae. Okay, and they have what? They have trachea to breathe. Compound eyes, 360. What else? They have 2 pair of wings, 3 pairs of jointed legs. Yes, you can talk about their bodies divided into 3 parts. 3 parts. Okay, so this is all about insects. Now looking at the other arthropod, that is crustaceans. Okay, crustaceans. Okay, crustaceans. They have how many pairs of jointed legs? They have 4 or 5. Okay, remember, 4 or 5 pair of legs. Okay, legs. I'm so sorry, I'm writing it very fast. Okay, then there are 4 or 5 pairs of joint legs. They have 2 pair of antennae. 2 pair of antennae. Okay, and you can also remember that their exoskeleton is calcified to form carapace. Okay, then having a look at arachnids. Arachnids, they have how many pair of antennae? They have no antennae. They have 0 antennae. Okay, then another thing you should remember about them is that they have 2 body parts. Cephalothorax. Okay, their head and thorax is joined to form cephalothorax, and they have abdomen. For the insect, it was head, thorax, abdomen. Here they have 2 body parts. Their head and thorax are joined from cephalothorax and abdomen. And how many pairs of jointed legs? They have 4 pair of jointed legs. 4 pair of legs. Okay, now having a look at the myriapods, they have many body segments. Okay, the other one is myriapods. Okay, they have many pairs of segments. They have many pairs of they have 1 pair of antennae. Okay, remember this, 1 pair of antennae. Okay, and they have what? They have many they have many body segments. They have many pairs of jointed legs. Okay, so the examples are centipede, millipede. For arachnids, you can give example as spiders, scorpions. Okay, and then for crustaceans, crabs, lobster, woodlice. Okay, and insect, of course, you know, cockroaches and all, you know, they can come in insects. Okay, now moving on, next we have what? We have vertebrates. Okay, invertebrates, first of all we have fish. Okay, fish, amphibian, reptile, bird, mammal. Okay, now in fish, they have scales. Okay, they have gills. They have what? They have fins. Okay, they have fins, and they have no, what do you say? No, they lay eggs. Okay, eggs. Eggs. Okay, eggs with no shells. With what? With no shells, and they lay their eggs in water. Okay, so they have scales, they have gills, they have fins, the eggs have no shells, and the eggs are laid in water. Then we deal with amphibians. Okay, amphibians, the adults live in land and the young live in the water, larvae. Okay, so you should be knowing that. Next, what do we have to say? Yeah, they have no scales again. Okay, they have scales. Fish have scales. Amphibians have no scales. Scaleless skin, very commonly asked in MCQ. Okay, and another thing that you have to know is that their eggs, no shell. No shell. And yes, their eggs are also laid in water. Okay, their eggs are laid in water. Next, another thing that you have to know, is that the adults have lungs, but the young ones, they have gills for gas exchange. Okay, this is something you should be knowing. Okay, next, and yeah, they have moist skin. Okay, and their skins have no scales. Okay, now next you will be dealing with reptiles. They have what type of shells? Rubbery shells. Okay, they have rubbery shells. Only reptiles and birds, they lay eggs with shells, whereas fish and amphibians, they have no shells for their eggs. Okay, they have rubbery shells. Then what else? They have dry scaly skin. They have a scaly skin. Okay, amphibians, they do not have scales on their skin. Okay, that's all you need to know for reptile. Examples if important can be lizard, crocodile. Okay, then next, snakes, which you're always surrounded by. Okay, birds. What about birds? They have what? The distinctive feature of birds. First, let's discuss the distinctive feature. The beak is the distinctive feature. They don't have teeth, they have beak. And then the distinctive feature, which sets them apart from everything else, is that they have feathers. Okay, and then they have, what do they have? They have wings. Okay, and they might have few scales on legs. Okay, and they lay eggs with hard shells. Okay, these two are distinctive features. Okay, here, hard shells. Okay, so this was all about vertebrates. And another vertebrate, mammals. Okay, mammals have many distinctive features. What is one distinctive feature? Fur or hair. Okay, they will give you a picture and they'll tell what is an evidence that this is a mammal. You can say fur or hair. Okay, fur or hair. And even external ear, pinna, okay, whatever you say it. Okay, next, another thing about mammals is that they have their young ones develop in the uterus. Okay, they feed milk to their young ones. Okay, and they have sweat glands. Okay, and they have 3 types of teeth. Okay, 3 teeth. Okay, another thing that you can add here is that yeah, you can add about the diaphragm and that. Okay, and another thing, okay, another thing that I want you all to know is that birds and mammals, they are basically called warm-blooded or homeothermic because they can maintain their own temperature, whereas the other ones, reptiles, amphibians, and fish, they cannot maintain their temperature and they are called basically cold-blooded, that is poikilothermic. Okay, now moving on. Okay, this we have discussed. Now moving on to, okay, let me erase all this. Now moving on to, okay, this was the animal kingdom. This was all about the animal kingdom. Now moving on to the, oh my god. Okay, now moving on to the plant. Okay, moving on to the plants. Plant. Okay, even plants are divided into 2 parts. There are ferns. Okay, there are ferns and there are flowering plants. Flowering plants. Okay, then ferns. Okay, first let's discuss ferns. Ferns, basically they do not produce flowers. Okay, no flowers. Okay, then how do they reproduce? They reproduce by forming spores. Okay, they reproduce by spores. Spores they use to reproduce. Okay, and they have spores basically on the underside of the leaf. Okay, underside of leaf. Okay, underside of leaf. Okay, sorry, you have to deal with my handwriting. And then next, okay, they do not produce flowers and they have roots, stems, and leaves. Okay, ferns are pretty simple to remember. Just know that their leaves are called, okay, and yeah, one thing, okay, their leaves, I'll just start it here. Their leaves are called fronds. Okay, remember this, fronds. Okay, then next, dealing with flowering plants. Okay, I'll just erase this. Next, dealing with flowering plants. Flowering plants are again of 2 types. They're again divided into 2 types. Dicots. Okay, dicots and monocots. Monocots. Okay, first of all, we'll be dealing with them separately. Let's deal with dicots over here first. Or let's just make a column so that it's easier for you all to remember. Okay, we have to be very quick. Okay, let's just do it here. Okay, first of all, dicot and monocot. Okay, so what's the difference between them? Basically, they are cotyledons and the seed. Okay, and they have 2 cotyledons whereas they have 1 cotyledon. What's the spelling? Wait, cotyledon. Okay, cotyledon. Then they have 1 or 2 cotyledon. Then next, dicot, they have flower pods in multiples of 4 or 5, and they have flower pods like flower petals. Okay, in multiples of multiple. Okay, remember it's a multiple, so it can be a 3, 6, 9 like that. Okay, multiples of 3, and they have flower pods in multiples of 4 or 5. Okay, I'm not writing full because of course we don't have so much time. Okay, what the hell? All right. Okay, then next you should know that they have a network of, sorry, my man. Okay, network of branched veins. okay, of branched veins, okay, and they have parallel veins. Make sure you see a picture of them, okay, so because you can be asked to identify as well, okay. And then the dicots, basically they have a taproot system, okay, taproot, whereas monocots, they have a fibrous root system, okay. Fibrous root is basically they have you can say that their roots grow like, you know, directly from the stem. All right, then next you should be knowing that their vascular bundles are arranged in ring, okay, and their vascular bundles are arranged randomly, okay. Then yeah, I think that's all about it, okay. Now let's move on to the next part, okay. Now we have dealt with animals, we have dealt with, what do you say, plants, okay. Now the next one, what we will be dealing with is basically, yeah, what are we going to be dealing with? Yeah, we're going to be dealing with fungus now, okay. Let's deal with fungus now, okay. So basically when we talk about fungus, in fungus they have a cell wall, okay. Fungus, remember that they have hyphae first of all, okay. They feed using their hyphae and they reproduce by spores, okay, and hyphae for feeding, okay. So basically they spread in the food and that's how they get their, you know, food, okay. The next thing about fungus is that they have cell wall, okay, and their cell wall is not made of cellulose like in plants, okay. I don't know if I mentioned this, but in plants the cell wall is made up of cellulose, but in for fungi, sorry, it's not made up of cellulose, it's made up of some other substance that's called chitin, I believe, okay. Then they do not have chlorophyll, so they cannot make their own food, right? If you have chlorophyll, then only you can make your own food. So how they make their own food is that they feed saprophytically, so, or parasitically, okay. So basically fungus, okay, and that it feeds saprophytically, and saprophytically means that it takes its nutrients from dead organisms. And by the way, fungus can be multicellular or it can be unicellular, or it can be unicellular, okay. So it can be any. And parasitically, by the way, means that it relies on its host and it basically harms the host, okay, and it gets the benefit, all right, okay. Now next, oh my god, even the highlights are on, okay. Now next we will be dealing with the prokaryotes, okay. Prokaryotes are basically bacteria. I'll be saying it now rather than writing it so that we don't waste much time, okay. Basically prokaryotes, they have a cell wall, they have ribosomes, okay, and their cell wall again is not made up of cellulose, and their cell wall is made up of peptidoglycan, all right, and they have ribosomes, all right, but they do not have mitochondria, okay. And the main thing about prokaryotes is that they are unicellular, most of them, and they don't have a nucleus, they have a loop of DNA, and they also have plasmids, okay. So these are some main things about prokaryotes, okay. Next we'll be dealing with protoctist. Protoctist is pretty straightforward because basically some protoctists have chloroplast, some have cell wall, some don't, some are multicellular, some are unicellular, some are like animal type and some are plant type. So there's not pretty much to learn about protoctist, okay. Now next, moving on, we have basically completed all this, okay. Let's discuss virus. Basically virus is not categorized in any of the kingdoms of the living organism because it does not show any of the characteristics of the living organism. The thing is that it basically, yeah, it does not show any characteristic of the living organism. It basically consists of a protein code with the genetic material inside, okay. I'll try to show it to you. This is the protein code and you can say this is the genetic material inside, okay. And it only works, okay, when it is inside a living organism. So basically yeah, it is not categorized in any of the 5 kingdoms, and it's basically a separate thing you have to study, and it's only for extended, by the way. You all can see, you know, whenever it's here it's extended, and whenever it's here it's like, you know, good. Okay, now next about the cell structure, dealing with the cell structure, okay. So basically cell wall first. Cell wall is basically made up of cellulose, which belongs to polysaccharides, okay. Next, cell wall, it resists the turgor pressure, all right, and it prevents bursting, and it's basically allows most of the substances in, but it is a cell membrane, okay. This is a cell wall and it is a cell membrane inside which controls what enters or leaves the cell, okay. Okay, so it is a cell. The job of the cell membrane, it is to separate the contents of cell from environment and to control basically what enters and leaves the cell. Next, about the cytoplasm. Okay, no, actually it is, okay, let's discuss about the nucleus. Basically of the nucleus, Nucleus is like a storage of DNA. It contains the DNA. It controls all the activities of the cell. It produces RNA, okay, and it makes ribosomes, okay. Then it controls most of the functions of the cell, okay. It contains the DNA, yeah, okay. Then next, talking about chloroplast, talking about cytoplasm. Okay, about cytoplasm, it is a site for most of the, it is a site for all the reactions in the cell, okay, and it is mostly made up of water, and most all the metabolic reactions take place here. Then about chloroplast basically it contains the chlorophyll, and chlorophyll absorbs energy from light and converts it light energy into chemical energy for the synthesis of the carbohydrates. Then dealing with ribosomes are basically used in protein synthesis, okay. They're used to make protein. Next, dealing with mitochondria releases energy from respiration. Respiration basically takes place in the mitochondria, and it makes ATP. Then vacuoles basically contain cell sap, and that cell sap is basically substances, okay. And plant cells, they have a large permanent vacuole, whereas animal cells, they have smaller vacuoles, and they are called vesicles, and they basically contain cell sap. And what is cell sap? Cell sap is a fluid that fits in the vacuole, all right. Now moving on, next we have the structure of a bacterial cell, okay. And by the way, let me tell you, cell wall, as we discussed before, cell wall and chloroplast is not present in, out of all these, cell wall and chloroplast are not present in animal cell. It's only present in plant cell. Rest all is present in animal cell, except that, you know, vacuoles are smaller in animal cells. They are present, but they are smaller in animal cells, and they are called vesicles for animal cells, and the plant cells, they have a bigger vacuole in the middle. Alright, so if you draw a plant cell, it's basically like this in the middle, a large vacuole, okay. So next, describe the structure of bacterial cell. They have a cell wall, cell membrane, cytoplasm. We have already dealt with it, all right. They have plasmid, they have circular DNA, okay. Then you should be able to identify this entire class. Make sure you do that well. Then the basically specialized cells, ciliated cells, movement of mucus in the trachea and bronchi, okay. They basically, remember, they're present in the trachea and bronchi. Root cells absorb mineral ions and water. Passive mesophyll cells are used for photosynthesis, and they contain lots of chloroplasts. The neuron is for conduction of electrical impulses, and it's in the nervous system. Red blood cells are in the blood, and they are for transport of oxygen, okay. Hemoglobin combines with oxygen to form oxyhemoglobin. Sperm and egg cells are for reproduction, okay, and the sperm are in the testes and egg cells are in the ovaries. Okay, next, describe the meaning of cell, tissue, organ system. This is written in order, and cells is basically like you can say it's basically a cell, all right. And tissue, it's the smallest unit of a living organism, you can say, for cell. Then tissue, it is a group of similar cells working together to perform a function. Organ is a group of similar tissues working together to perform a function. And organ system is basically all of the organs working together, like circulatory system, okay. In circulatory system, you can say that the heart is an organ, all right. The blood is a tissue, okay. A red blood cell is a cell, all right. So they all work together to perform a function, all right, and then they form an organism. All right, magnification formula, very important. Image size divided by actual size. If you have studied physics, you'll be able to do the conversions very well. Okay, the centi is 10 to the power minus 2, milli, micro, nano. You've all studied physics, so you all must be knowing this, all right. Okay, anyways, I will tell you, if you have to convert between millimeters and micrometers, if you have to convert micrometers, so see centi, milli, micro, so use 10 to the power of minus 6. Okay, this is what you'll be using for micrometers to convert it to meter, okay, to convert it to meter. And this is for millimeter if you have to convert it to meter, okay. So next, okay, next fast. Okay, diffusion is the net movement of particles from a region of the high concentration to a region of the low concentration as a result of the random movement, okay. The energy for diffusion, where does energy come from? It comes from the kinetic energy of the random movement of the molecules or ions. It comes from where? The kinetic energy, okay, very important. From high region, they go to the lower region until they are evenly spread as a result of the random movement. The energy is due to the kinetic energy of the molecules or ions. Some substances move into and out of the cells by diffusion through the cell membrane, through which the cell membrane. All right, now what is the importance of diffusion in a living organism? What are the importance of diffusion? Basically, diffusion happens where? Okay, so basically diffusion happens in the small intestine. Small intestine. Where does diffusion happen? You will learn it later. That digested food like glucose, amino acids, fatty acids, and cholesterol, okay, the digested food, the glucose, fatty acids, amino acids, and cholesterol, they move from the lumen of the small intestine, they move to the villi, okay, then it happens in the leaf, the oxygen and carbon dioxide diffuse into and out of the leaf, okay, water vapor as well, then it also happens in the lungs, okay, it also happens in the lungs. So make sure you know all of this. These are the uses of diffusion in living organisms. Okay, where does the energy for diffusion come from? We've already discussed. Investigate the factors that influence diffusion limited to surface area. Greater the surface area, greater the diffusion. Greater the temperature, greater diffusion. Greater the concentration gradient, greater diffusion. Lesser the distance, more the diffusion. Now, okay, so basically water, it has a role in digestion, in excretion, in transport. How? In digestion, basically all the enzymes are present in water. Okay, all the enzymes are dissolved in water. Then talking about excretion. In excretion, the kidneys excrete urea, and that urea is dissolved in water to form urine. Next, in transport, the blood, it is made up of the blood plasma. Basically, around the body, the substances which are transported around the body are dissolved in the water in the blood plasma. Okay, the cytoplasm is made up of the water. Then next, what else? The digestive food moves, the digestive food in the alimentary canal need to be moved to cells all over the body, and the water acts as a solvent over there. Alright, dissolved substances can easily be transported around in organisms, like for us in the blood, for plants it can, in the xylem transports it. Alright, so it is a very important solvent for our body. Okay, so next, you should remember that water diffuses through partially permeable membrane by osmosis. Okay, these three are the uses of water. Next, water diffuses through a partially permeable membrane. Which is the partially permeable membrane? Okay, so the partially permeable membrane is the cell. Okay, so the partially permeable membrane is the cell membrane. We have discussed before. Cell wall allows almost everything to pass through, but cell membrane is the one that controls what should enter and what should go out. Okay, it's written here. Water moves into and out of the cells through the cell membrane. Okay, cell membrane allows smaller substances to pass through, but not bigger ones. Okay, what is osmosis? The definition. Okay, it's a net movement of water molecules from a region of their higher water potential to a region of the lower water potential. Okay, through a partially permeable membrane. Okay. So remember, osmosis deals with water, whereas diffusion is just for the particles. Okay, it's just about the particles. Like in the small intestine, you about the food particles like glucose, etc., oxygen, carbon dioxide, because that are all gases. Okay, we deal with water in osmosis. Remember, even water vapor comes in diffusion. Okay, it moves out into the leaf, etc., by diffusion. Okay, osmosis is about water, okay, from a region of high water potential. Remember, dilute solution. Okay, dilute means that more water, more H2O, and less of solute. Okay, and concentrated means that more of solute and less of water. So it moves from a region where there's more water to a region where there's less water. All right, now I want you to remember that it is through a partially permeable membrane, because this is very important. Diffusion does not require a partially permeable membrane. Osmosis does require a partially permeable membrane. And we will see later that even active transport requires a partially permeable membrane. These are three ways in which substances move into and out of a cell. That is diffusion, osmosis, and active transport. First, let's have a closer look at osmosis so we can move on to active transport. Okay, so you should be knowing that plants are supported by the pressure of the water inside the cell pressing outwards on the cell wall. The reason of this is that, first let's look at this. If we place a plant, okay, if we place a plant cell in a solution, okay, in a solution, or in, you can say any solution, yeah, this is a plant cell, okay, and the plant cell has, and just draw it bigger, okay, the plant cell has more water and the outside has less water. So you will say that the plant cell is dilute and the outside one is concentrated. Concentrated means this has more solute. Okay, this has more solute. So what is going to happen? What is going to happen? We're dealing with water. Water will move out of the plant and into the solution because it moves from high to low. What is going to happen now is that the cell membrane will pull away from the cell wall. Okay, the cell membrane will pull away from the cell wall. So here, plant cells are placed in a concentrated solution. Water will move out of the plant cells by osmosis, right? We saw it here. It will move out of the cells by osmosis. If the plant cells become flaccid, now they're going to become flaccid. The cell membrane will pull away from the cell wall, okay, and the vacuole will shrink. okay, and it will cause it to become flaccid, and eventually the plant, okay, the term that you use is flaccid, okay, flaccid, and eventually it will become plasmolysed, okay, so eventually it will become plasmolysed, okay, so in that time, okay, so when it becomes plasmolysed, basically the cell membrane has pulled away from the cell wall. The cell membrane has pulled away from the cell wall. The cell membrane has pulled away from the cell wall, and the vacuole has shrinked. Okay, so remember this. Next, what will happen if we place a plant cell in a solution that is dilute and the plant cell is concentrated? That is, this has less water and the outside has more water. Okay, and this, what do you say? This is, okay, this is more water, means it is dilute, okay, that inside of the plant cell is concentrated. So what is going to happen? It will move from outside to inside of the cell. The water will move from outside to inside of the plant cell, and the plant cell will become turgid, and the cell membrane will pull away from the cell wall will press on the, see, you can see it here, this supported by the pressure of water inside the cells pressing outward on the cell wall. So the cell wall will press outside towards the cell, and the cell will become more turgid. All right, because this is because the water molecules push away the cell wall, okay, they push the cell membrane against the cell wall, and they will increase the turgor pressure, and the plant will become turgid. The plant cell will become turgid. So more water is basically always good for the plant cell than less water, because it can cause the plant cell to die. Okay, can cause it to die. Now, dealing with animal cell, what is going to happen if we place an animal cell? Okay, if we place an animal cell in the same thing. First, let's deal with this has more water than outside, and this has less water. So even in the plant cell, it will lose water and it will become shriveled. Okay, it will lose water and it will become shriveled, and they will basically, because the effect of the losing the water will be very much on the animal cell. Okay, then next, if you deal with what is going to happen if it's placed in more water and this is less water, basically in a dilute solution, if it is placed, what is going to happen? Water will move into the plant cell, but because the animal cell, but because the animal cell does not have a cell wall, it will cause the animal cell to blast. Okay, it will cause the animal cell to blast, and the animal cell will die. And this, if this has a greater, the results of this is greater on animal cell of more water. Placing it in more water is greater, the effect of it is more greater on animal cell than plant cell, because animal cells don't have a cell wall, so it is not going to help. It's basically just going to blast the animal cell, okay, because they have no cell wall to create turgor pressure, and it bursts. Okay. Now, what is active transport? Active transport is the movement of particles from a region, okay, through a cell membrane from a region of their lower concentration to a region of their higher concentration, and this happens against a concentration gradient, and it uses energy from respiration. Remember, for diffusion, the energy was coming from the kinetic energy of the random movement of the molecules or ions, and the same thing for osmosis. But here, the energy for active transport comes from respiration, which happens in mitochondria, as we discussed before. Okay, in mitochondria. Okay, so this is where we get the energy from. The active transport is basically very simple, but remember, it's the opposite of diffusion and osmosis. It happens from lower to higher, and it is against a concentration gradient, not down a concentration gradient like over here. Okay, it's not down a concentration gradient, it's against a concentration gradient. Okay, and it uses energy from respiration, very important. Remember that active transport always requires a cell membrane, and so does osmosis always require a partially permeable membrane. Diffusion does not require a membrane, because you can see it this way. You can imagine this way, that when you spray perfume in the room, it diffuses from a region high concentration to low concentration, and it's eventually spread in the room. So was there a cell membrane over there? No. So diffusion does not require a cell membrane. Now, what is the importance of active transport? Basically, the root hair cells, okay, the root hair cells, they take in ions from outside of the plant by diffusion, by active transport. See, the root, okay, so these are the roots, all right, and what is going to happen is that there are more ions, like there are lesser ions outside, like you can imagine Na plus, sodium, okay, and, okay, now let's not take sodium, okay, so let's imagine there is K plus. Okay, there's less over here and there's more in the root. Okay, so the root needs more of it. Okay, it needs all of it. It needs like most amount of the potassium, most amount of the potassium. Okay, so what it's going to do is that it's going to absorb it by active transport. What the hell just happens? Okay, so it's going to absorb it by active transport, and it's going to take it against the concentration gradient by active transport. Okay, I don't know what happened. Okay, anyways, so it's going to take it by active transport, and another importance of it is that it's the uptake of glucose by epithelial cells in the villi of the small intestine. Okay, and then by the kidney tubules. Okay, in nephron they reabsorb if you remember, they reabsorb all of the glucose, most of the water. Okay, we will be discussing it later. Okay, so remember there are three uses: by root hair cells, then in the epithelial cells, okay, and the other one is in the, what do you say, in the kidney. Okay, and kidneys, okay, whatever, kidney. So kidney, epithelial cells, uptake of glucose by the epithelial cells in the villi, and the other one is the root hair cells for osmosis. Remember the uses of water also. It is in digestion, excretion, and in transport. Okay, so now we are done with all this. Okay, and remember that, okay, I forgot to talk about carrier proteins. Carrier proteins, they basically move the molecules across the membrane during active transport. They are present in the cell membrane and they change the shape using energy from respiration, of course, and they move the molecules into and out. Now next, discussing biological molecules. Okay, also remember that the carrier proteins, okay, the carrier proteins are specific. Okay, they are specific. Okay, so now we will be discussing some three chemical elements, that is carbohydrates, fats, and proteins. Oh my god, what the hell? Carbohydrate. Oh my god, why is this so annoying? Okay, carbohydrates, fats, and proteins. I want you to remember that all of them are made up of carbon, hydrogen, and oxygen. But protein is also made up of, protein is unique and including carbon, hydrogen, and oxygen. Okay, so protein is also made up of nitrogen and a bit of sulfur, sometimes sulfur too. You do not. Okay, so it contains sulfur but only in less quantity, but mostly in our syllabus they mostly talk about nitrogen, so make sure you know that. Don't say that protein only contains nitrogen. They have carbon, hydrogen, and oxygen. These are elements that are present in all carbohydrates, fats, and proteins. But proteins also have nitrogen with them. Alright, so I hope you got it. Next, you should be knowing that large molecules are of course made from smaller molecules, and starch, glycogen, and cellulose are made from glucose, proteins are made from amino acids, and fats are made from fatty acids and glycerol. Remember, starch, glycogen, and cellulose are carbohydrates. Starch, glycogen, and cellulose are carbohydrates, and they are made up of glucose, which are smaller molecules, proteins from amino acids, and fat and oil from fatty acids and glycerol. So basically each fat molecule contains contains glycerol and 3 fatty acid molecules that are combined to it. Okay, so 1 glycerol and 3 fatty acids. You don't need to know all that, but just know it's made of fatty acid and glycerol. Number is not needed. Okay, now we will be discussing the tests. Okay, so this is basically a very small and sweet chapter, okay, but it requires a lot of focus. Okay, now the tests, they're mostly asked in paper 6, but they can also be asked in MCQ. Iodine solution is a test for starch. Basically, it turns from orange-brown to blue-black if starch is present. All right. Next, we have Benedict solution. Okay, so you can write here, it turns... Okay, I'll just discuss it. Writing is going to take a lot of time. Okay, Benedict solution. Benedict solution is used for reducing sugar. Basically, in Benedict solution, you heat it in a water bath. You take Benedict solution. Okay, with the solution. Okay, so Benedict solution is used for reducing sugars. You heat it in a water bath up to 80 degrees Celsius, and then Benedict solution undergoes through a range of colors, like it turns orange red, and it can also go green, yellow, orange to brick red. Okay, so depending on the concentration of the sugar, then it goes through a range of colors. Okay, biuret for proteins. Please make sure you know all these names. Okay, memorize them. Okay, use some acronyms or something and memorize them. Okay, for biuret for proteins. Who can tell me? Yes. Okay. So biuret reagent you use to test for protein, and it's a blue solution, and it turns purple when it is mixed with protein or amino acid. So it is from blue to purple. Okay. For iodine, it is from it turns blue-black from orange-brown. Benedict solution goes through range of colors from green, yellow, orange to brick red. Okay. Then ethanol emulsion test is used for fats and oil. Basically, the thing is that fats and oil, fat is a lipid, okay, they're both lipids, okay, but fat is a lipid that is solid at room temperature, and oil is the lipid that is a liquid at room temperature, okay. And now what the thing about them is that they are, okay, they do not dissolve in water, okay, they do not dissolve in water, but they do dissolve in ethanol. They do dissolve in ethanol. So we use this to check if they are present or not in any food. So what we do is that first the food is shaken with ethanol, okay, to allow for any fats in it to dissolve in the ethanol. Then what you do is you pour it in a clean test tube containing water. So first you shake it with ethanol, then you pour it in a clean test tube containing water. And if fat and oil is there, fat or oil is there in the food, okay, then you will see tiny droplets form in the water, which will give it a milky appearance. Okay, which will give it a milky appearance. Otherwise, it will just stay the way it is. Okay, and okay, they'll form tiny droplets. Okay, and yeah, this is called the ethanol emulsion test. Okay, next for DCPIP for the vitamin C. DCPIP, how do you do this? Okay, so DCPIP for vitamin C, it basically changes from purple to colorless. What you do is basically you take vitamin C, okay, like maybe lemon juice or something, and then you take DCPIP and you add drops of it, okay, or yeah, you add drops of it and you see how long it takes for it to decolorize, okay, and the number of drops and all that, okay. That's all a paper 6 thing, but yeah, just know that the color changes for now. So DCPIP, it changes from purple to colorless. Ethanol emulsion test, it gives you a milky appearance. If not, it just remains like that. Test Biuret reagent, it turns from blue to purple. Yeah, and then Bendix solution range of colors, blue to orange, yellow, green, yellow, and all that. I already told you. Okay, iodine test, basically from orange, brown to blue, black. Now, okay, so basically the glucose is the way that carbohydrates are transported around the body. Okay, they're transported around the body, and the way that the glucose is stored, okay, the way the glucose is stored in the human body is through is by glycogen. It is stored in the form of glycogen. Okay, so the liver basically does all that. The liver and pancreas to work together to do this, and it is stored in the liver cells. Okay, and it is stored in the form of starch for the plant cells. Okay, now, next, another thing that you must be knowing is that cellulose and both starch, the cellulose basically, yeah, the cellulose is made up of glucose molecules, right? So in the cellulose, the way they link up, okay, they form straight lines and they don't coil up in a spiral shape. Okay, they don't coil up in a spiral shape, they basically link up in straight lines. Okay, whereas for a starch molecule, they form a spiral shape. Okay, now let's discuss some uses of fats. Okay, fats are used in making cell membrane. Okay, cell membrane. Okay, next they are used in storage, for storage and to provide energy. I think this comes later on though, but we'll just discuss it here. Okay, for storage, and they're used to provide energy. Okay, and they're used for insulation. Okay, they keep you hot, all right, the layer of fat on you, and it's an energy store, okay, store of energy, all right, it is a storage material, and yeah, that's it, okay, and it's for insulation. Now next with proteins, okay, proteins also, they have many uses, okay, I'm just writing it here so only, okay, this is for fats. Now let's discuss protein. Okay, protein, they have many uses. Okay, when looking at protein, they have so many uses. First of all, enzymes. Okay, we'll have a look at it in a bit. Enzymes, then they are used in cell membrane, again, like the fats. Okay, they are used in keratin. Okay, well, what is keratin? Basically, it's found in the hair and the fingernails. Okay, it's a protein. Then it is used in antibodies. Then it is used in the carrier proteins we just discussed over here before in like active transport. Okay, carrier proteins, then collagen, which is found in skin and other tissues and keeps the tissues in good repair. Okay, and it is also in growth and tissue repair. Growths and tissue repair. Okay, so these are some uses of protein. Okay, so it is enzymes, cell membrane, collagen, keratin, hormones. Okay, yeah, hormones, I think I missed that. Yeah, hormones. Okay, hormones. And it's also in hemoglobin, by the way. Hemoglobin. Hemoglobin. Okay, I don't care. Okay, hemoglobin, antibodies, enzymes. Okay, all these are the uses of protein. Okay, so these all are made up of protein. These all are made up of what? These all are made up of proteins. Okay, now let's have a look at the DNA. The DNA, this question I believe was asked in paper 4. Anyways, okay, two strands coil up together to form double helix. Two of the strands, they coil up together. Okay, there are two strands. Wait, yeah, there are two strands and they coil up together to form a double helix. Okay, and each strand contains chemical called bases. A with T, and remember A with T. Okay, they pair up like this: A with T, and it is C with G. Okay, each strand contains bases. Okay, like this will have bases. Okay, this will have base, this will have a base, this will have a base. Okay, and they'll pair up. Okay, so you can imagine this as like two strands. Okay, then as we discussed before, each strand contains chemicals that are called bases. Okay, we discussed it here. Now so each strand contains chemicals that are called bases. Another thing that you need to know is that the bonds between the pair of bases hold the strand together. The way that A bonds with T and C bonds with G, adenosine with thymine, okay, this all you don't need to know the names, by the way. Okay, so these hold the strand together. Alright, then the other thing is that the bases always pair up in the same way, A with T and C with G. Okay, this was pretty simple and full names are not to be known. Alright. Okay, now moving on to catalyst. A catalyst is basically a substance, okay, that increases the rate of chemical reaction and it is not changed by the reaction, okay. It is basically added in reactions and it increases the rate of the chemical reaction and it is not changed by the reaction. You all must be knowing this because especially if you are... Okay, so catalyst is a substance that increases the rate of reaction and it is not changed by the end of the reaction. Okay, so enzymes are proteins that are involved in metabolic reactions where they function as biological catalysts. Okay, as we discussed before, enzymes are made up of protein. Okay, so that are involved in metabolic reactions. Okay, metabolic reactions that take place inside the cell and they function as biological catalysts. Now, why enzymes are important in all living organisms in terms of reaction rate necessary to sustain life. So basically, we want to know why enzymes are important. Okay, why are enzymes important? The reason is that without enzymes, basically the reactions would take place too slow to even sustain life. Okay, they would take place too slow to sustain life. So I'll write it here: reactions. Okay, reactions too slow to sustain life. All right, too slow to sustain life. Okay, to sustain life. Okay, now enzyme action with reference to the shape of the active site of an enzyme being complementary to its substrate. Okay, so basically in an enzyme, okay, just imagine this is an enzyme. The shape, this is an enzyme. Okay, this is an enzyme. Okay, and this is the active site of the enzyme. Okay, it is in the active site that the substrate molecule will fit. Okay, so the substrate is going to look like this, and this is the substrate. Okay, so remember that the substrate and active site are complementary. Okay, complementary means they have the same shape. Complementary. Okay, so they're complementary. So you can like see here. Okay, so this one is going to fit over here and they're complementary because they have the same shape. Basically, the shape of the enzyme was like this. The triangle is not going to fit here. So this enzyme will not work for the substrate. Alright, so enzyme is also specific for a specific substrate. Alright. Okay, and when the substrate binds to the enzyme, this is called the enzyme substrate complex. Okay, it's called the enzyme substrate complex. Okay, so you all should be knowing that. Okay, enzymes work best at an optimum temperature. Okay, enzymes work best at an optimum temperature. Okay, like if you draw a graph, first the activity of enzyme will increase, then it will reach the maximum and then it will decrease. Why it will decrease is that this is called the optimum temperature at which enzyme works best, and when it will decrease, that is because the shape of the enzyme has changed and the enzyme has denatured. Okay, and over here the enzyme is not denatured. Remember that. Okay, over here the shape of the enzyme is not denatured. Only the kinetic energy of the enzyme is low, and the enzyme and the substrate are moving at slow speed, so there are less chances of them colliding and resulting in a reaction, which is why the rate of reaction is lower over here. Okay, and change in pH, any change in pH. Okay, optimum, basically what is optimum temperature? Optimum temperature is the temperature in which the enzyme works the best. Okay, and optimum pH is the pH in which the enzyme works the best, the maximum rate of reaction. Basically this is, okay, this is time, okay, and this is the rate of reaction. Okay, this is the product rate of reaction. I think right over here. Okay, it's actually the rate of reaction. Okay, it's actually the rate of reaction. Okay, rate of reaction, and then when the optimum pH is reached, it's the maximum rate of reaction. Okay, and even the maximum optimum pH, optimum temperature, that is when it works the best, and it is at this point. Okay, for any pH or temperature graph, and it's different for every enzyme. But the thing is that you should remember any change in pH, low or high pH, denatures the enzyme. I'm going very fast because this is revision. Okay, and any change in temperature, any change, lower temperature does not denature the enzyme, but only higher temperature denatures the enzyme. But lower pH and high pH both will denature the enzyme. Okay, I hope you got this. Now let me just quickly tell you some reactions which what you must be knowing about enzyme. Please listen carefully. Okay, so hydrogen peroxide is broken down by catalase to form water and oxygen. Starch is broken down by maltose to form amylase. Maltose is broken down by maltase to form glucose, and this glucose is used to make cellulose. Then you can say lipases breaks down carbohydrates, that is fats and oils. Lipases breaks down fats and oils, that are lipids, to fatty acids and glycerol. It is the carbohydrates that breaks down maltose is called maltase, okay, and the carbohydrates that breaks down sucrose, as I told you before, is amylase. Okay, hydrogen peroxide is broken down by catalase to form water and oxygen. Starch is broken down by maltose, amylase to form. Starch is broken down by amylase to form maltose. Maltose is broken down by maltase to form glucose, which is used to make the cellulose, and lipases break down fat and oil. Okay, now, I want you all to know that denaturation is irreversible. Means once an enzyme is denatured, it cannot be used again. So it is a big disadvantage if an enzyme gets denatured, and which is why even our body temperatures are controlled for enzymes to work best. And we will see later how they are controlled. It comes under the topic that is homeostasis. Alright, and as I told you, low temperatures do not denature the enzyme. Remember that as well. And the more the energy the molecules have, the faster they will move, and the more collisions that will happen, as we discussed before. Okay, now moving on to plant nutrition, which is a bit bigger than the topics we just did, like biological molecules and enzymes. Okay, basically photosynthesis is a process, it's a process by which plants synthesize carbohydrates using from raw materials, using energy from light. They basically take the energy from the light, and they have raw materials, and they from raw materials using energy from light. Alright, so what they do is that they make their carbohydrates from raw materials, whatever they have, and they use energy from light. Alright, so what is the reaction? Carbon dioxide plus water gives glucose plus oxygen in the presence of light and chlorophyll. So this is a necessity for photosynthesis to take place. Carbon dioxide plus water gives glucose plus oxygen. Glucose is the carbohydrate that is synthesized. Now next, chlorophyll. Chlorophyll is green pigment that is found in chloroplasts. Chlorophyll transfers energy from light into energy in chemicals for the synthesis of carbohydrates. Chlorophyll transfers. The chlorophyll is a green pigment that is found in chloroplast. In the chloroplast there is chlorophyll, and the job of the chlorophyll is to convert the light energy into chemical energy for the synthesis of carbohydrates. Now, this is a formula. You all should memorize it by heart. How to balance it. Basically, carbon dioxide plus water gives C6CO2 plus 6H2O gives C6H2O6 plus 6O2. Now next, basically add 6 in front of everyone except for glucose. Now, what are the uses of the carbohydrate that is made in photosynthesis? Must memorize. It is used as energy source, starch as energy source, cellulose to build cell wall. Then glucose is used in respiration to provide energy, because glucose plus oxygen gives carbon dioxide plus water plus energy in respiration. This is the opposite reaction. This reaction is basically the opposite in respiration. So sucrose for transport in the phloem, it is converted to sucrose for transport in the phloem, and there is a reason for that. Basically, the reason why why glucose is not transferred the glucose is not transferred in the form of glucose and is transferred in the form of sucrose is that sucrose contains more energy than glucose. And it is more efficient for both in transport and in storage, and glucose is highly reactive, so it is changed to sucrose for transport. Remember that. So then we studied before also, by the way, that starch, cellulose are both of them are made up of glucose. Anyways, sucrose is used for transport in the phloem. Even that we studied. Anyways, and nectar is used to attract the insects for pollination. Even that is made from the glucose sugar. So you can see how important the glucose is for the plant. Next, what is the importance of nitrate? Basically, nitrate, you should be knowing that nitrate is used for making amino acids, and magnesium ions are used for making chlorophyll. So basically, even the glucose is used to make the amino acids C6H12O6. But remember, amino acids will result in making proteins. So proteins also contain nitrogen, and that nitrogen they have to get from the nitrate ions, which will be in the soil. We will study more in detail in the nitrogen cycle how these nitrate ions come into the soil. Alright, so this is needed to make what? To make amino acids which will help in the growth. As we saw, protein helps in growth. Amino acids. So the amino acids will make proteins, and the protein will be needed for the growth. So without these nitrate ions, what is going to happen is that there is going to be weak growth. Okay, it will affect the growth of the plant. Now chlorophyll, of course, you know, it gives the green color to the plant, right? And even glucose is used to make chlorophyll, but chlorophyll is not a protein, but it still contains nitrogen, by the way. But anyways, we need magnesium to make chlorophyll. Okay, and if we don't have magnesium, it is going to cause yellowing of the leaves, because magnesium is used to make chlorophyll, and chlorophyll gives green color to the plant. And if magnesium is less, so the green color will not be there, and you can see it in the yellowing of the leaf. Okay, so remember, if nitrate ions are not there, it's going to cause leaf growth, and if magnesium ions are not there, it's going to cause yellowing of the leaf, especially between the veins. Okay, now of course this investigations paper says, okay, but I'll just discuss with it with you. Okay, so first of all, just remember that HCO3 indicator is used to check how much carbon dioxide level is there. Okay, so if this indicator is yellow, it shows that the CO2 is high. Okay, and if this indicator is red, it shows that the CO2 is normal. Okay, normal, that is equal to the atmosphere. And if it is purple, it shows that it is low. Okay, so CO2 is low. So this is something you should be knowing, because it can be asked in the MCQs as well. Okay, now moving on to the leaf structure, very important. Most of the leaves have a large surface area and they are thin. Okay, now the reasons for it. Basically, what the large surface area is going to do, that it is going to increase the rate of absorption of the carbon dioxide. Okay, larger surface area, greater rate of diffusion, right? So carbon dioxide can diffuse into and out of the leaf faster. For this, you will say into the leaf, because of course it is photosynthesis. Okay, so carbon dioxide, oxygen, you know, photosynthesis and respiration is always taking in the plant. Actually, respiration is always taking place in the plant, and then there's light, photosynthesis is taking place, and if there's no light, photosynthesis does not take place. Okay, so there the carbon dioxide can diffuse into the leaf, so it allows another reason is that increased carbon dioxide can diffuse into the leaf. Okay, and another reason is that large amount of sunlight, increased light can fall into the leaf. Okay, and can penetrate through the upper epidermis. Okay, we'll see that right now. And why are they thin? Thin again, decreased diffusion distance. Okay, decreased diffusion distance, so it will happen faster. Okay, the decreased diffusion distance, so increased rate of diffusion, and increased carbon dioxide, more carbon dioxide, and carbon dioxide, I mean more carbon dioxide, faster carbon dioxide can reach the leaves faster. Okay, now, okay, so this is the structure of the leaf, the upper epidermis, and then the parasite mesophyll tissue, then the spongy mesophyll tissue, then the lower epidermis. Here air spaces are there. Okay, here the air spaces is the lower epidermis, spongy mesophyll. Okay, and then vascular bundle, which is made up of xylem and phloem, and chloroplast, which allows light to fall, which carries out photosynthesis. Then guard cells also have chloroplast. Okay, and the functions of the leaf structure are that the upper epidermis, it is transparent and it is thin, and it allows the light to pass through. Okay, and then the, what do you say, the parasite, the upper epidermis also secretes a waxy substance that is thin and transparent, and it is called the cuticle. This creates a waterproof covering, and it prevents the water loss. And the plants which are in the desert areas, they have a thick the thick cuticle, because they can't lose water as there's less water available. Okay, then the next layer down is the parasitic mesophyll. The parasitic mesophyll contains lots of chloroplast, and it is column shaped, and it contains a large vacuole, so the chloroplast at the edge of the leaf, and okay, and they're tightly packed, and they contain lots of chloroplast, because the main function is photosynthesis, and they're close to the top of the leaf so that they get plenty of sunlight. And the transparent epidermis above them lets the light pass through to the parasitic mesophyll cell. Then talking about the spongy mesophyll cell, they have many air spaces between them, so that the gases can enter and leave, and it can allow them to reach through the parasitic mesophyll cell. Okay, then the guard cells. The guard cells allow the stomata to open and close, and they basically open and close, and they allow gas to enter and in and out, and that's their main function. And they close when the stomata has to be closed, and they open and the stomata has to be opened. Okay, so the vascular bundle now. The vascular bundle basically contains the xylem and the phloem, and it contains both the xylem and the phloem, and the xylem carries the water and the mineral, and water and minerals, and of course the phloem carries amino acids and glucose. And then the lower epidermis, and the chloroplast, of course, is the site of photosynthesis and contains chlorophyll, then which transfers energy from that into energy in chemicals. Okay, now the lower epidermis. The lower epidermis is basically it contains the guard cells. Okay, and it allows it to open and close, and sometimes this makes the cuticle, but usually it does not. Okay, so usually it does not. Okay, so this was all about the leaf structure, okay, the upper epidermis, okay, the air spaces. Then this is what we typically think of when we think of a plant cell, because it's just one of the types of the cell of a leaf. Yeah, because it's basically, there are many plant cells, but we basically consider this one is one of the type in the biocene. So that's all they're trying to say. Okay, so we have done how they're adapted, the labeling, everything. Okay, now moving on to the next part. So now moving on to human nutrition. What is meant by balanced diet? A balanced diet is basically a diet which contains all the required nutrients in the suitable proportion and the right amount of energy. Okay, so remember that. Okay, now what are the principal dietary sources and describe the importance of? Learn all of this by heart. Carbohydrates, fats and oils, proteins, vitamins limited to vitamin C and D, okay, mineral ions, fiber, water. Okay, so the principal dietary sources of carbohydrates, where you can get carbohydrates, are from wheat, okay, wheat, rice, maize and potato. Wheat, rice, maize and potato. And from where you can get the fats and oils is from cooking oil, meat, okay, cooking oil, meat, egg, dairy products, and oily fish. Okay, so that gives you fat and oily fish. Okay, and then proteins you can get it from nuts and seeds, peas and beans, okay, and also from fish, egg, okay, fish, egg, and also dairy products. Yeah, dairy products. Okay, so you have to memorize this. Okay, so now moving on to the uses of all of these. The carbohydrates, basically, they are used as a storage material. They're used as a storage material, as an energy store. We already discussed about fats and oil. Fats and oil is for insulation to make cell membrane as an energy store, and it basically provides mechanical production to the body, around the body organs. It is there, like the kidneys. And then the proteins are also needed in many places. We already discussed, like antibodies, hemoglobin, hormones, enzymes. Okay, so protein is very important. It's also used in making cell membrane. Okay, then vitamin C and vitamin D. Okay, so let's discuss it here. Oh wait. Okay, so vitamin C, basically you get it from citrus fruits. Okay, citrus fruits like lemon, oranges, okay, and raw vegetables. And if there's a lack of vitamin C, it will lead to scurvy, okay. And moreover, this scurvy, it is a pain in the muscle at joints which leads to the bleeding of the gums, okay, and it's bleeding of the gums and other places, okay. And the use of this, okay, you get it from this, okay. This is the disease you get if you don't have it. And the use of vitamin C is to make the stretchy protein collagen, which is found in skin and other tissues, okay. And even protein is used to make collagen, by the way. You discussed it before. Okay, collagen, which is found in skin and other tissues. So vitamin C, you get it from citrus fruits and raw vegetables. And the disease which you get if there's lack of vitamin C is scurvy. Scurvy is just a disease which causes pain in the muscles and the joints and bleeding in the gums. And collagen basically it is used to make, why is it needed? It is needed to make collagen, which is found in stretchy protein collagen. Yeah, it's a protein, of course, stretchy protein collagen found in skin and other tissues, and it keeps the tissues in good repair. Okay, so now vitamin D. Okay, now vitamin D. Okay, vitamin D, you get it from butter, egg yolks, and yeah, but basically most of our vitamin D is made when sunlight falls onto the skin. So vitamin D from butter, egg yolk. Okay, so butter and egg yolk. And so the use of this is that it helps calcium to be absorbed, for making bones and teeth. bones and teeth. Okay, so it is used for making bones and teeth, and the disease that is caused when there's a lack of vitamin D is called rickets. Rickets is a disease in which the bones become deformed and soft. Okay, so this is all about vitamin D. So basically, vitamin D you get it from butter and egg yolk, but most of it is made when sunlight falls on our skin, and then basically it is also used to. Yeah, the disease that it is used to make. The reason for vitamin D is that it keeps it helps calcium to be absorbed for making bones and teeth. And if you don't eat, if you have lack of vitamin D, you will get rickets in which the bones will become deformed and soft. So better eat vitamin D foods. Better eat butter and egg yolk. Okay. Okay. So now next is the hell. Okay. Next is calcium. Calcium. Basically, calcium you get it from. Okay, calcium you get it from milk and other dairy products. Okay, milk and dairy products, and even from bread. Okay, milk and dairy products, and even from bread. Okay, and what you get if you. Okay, well, where is calcium needed first? It is used for bones and teeth and for blood clotting. Okay, for bones and teeth, and it is also used for blood clotting. And basically, what happens if you have a lack of vitamin D is that you will get something that is called brittle bones and teeth. Your bones will become brittle. Okay, brittle bones and teeth, and you'll have a poor blood clotting. Okay, brittle bones and teeth, and you will have a poor blood clotting. So what you should do is you should what? You should drink milk and other dairy products, and you should eat bread. Okay, poor blood clotting. Okay, whatever. Now, next one which we have is very messy writing, I know. Okay, next one which we have is iron. Iron you get it from liver, egg yolk, green vegetables. Okay, green vegetables, and you also get it from. You also get it from what? From bread meats. Okay, from bread meats. Then why is it needed? It is needed to make hemoglobin. Hemoglobin. That's how I learned the spelling, hemoglobin. Anyways, it's used for making hemoglobin, which is the red pigment in blood which carries oxygen. Okay, and if you have a lack of hemoglobin, you'll get anemia. Okay, and in which they're not enough red blood cells, so the tissues do not get enough oxygen to them. So better eat liver and egg yolk and green vegetables and meat. Okay, now getting back to our syllabus. Okay, so the causes of scurvy and rickets, we are just discussed. Scurvy is caused due to lack of vitamin C, and the rickets is caused due to lack of vitamin D, just writing short forms. Okay, now in diagrams and images, you should be able to identify these diagrams we will discuss in the end. Okay, first of all, you should be knowing, we will discuss all the diagrams in the end, okay? Okay, first of all, you should be knowing ingestion is the taking of substances, example, food and drink into the body. Digestion is the breakdown of food. Then assimilation, after ingestion, first you take the food into the body, then it gets digested, that is, it gets broken down. Then the nutrients move from the intestine into the blood, okay, where it can be used up by the cells. So that is assimilation. There it is used up by the cells. And absorption is when it moves from the intestine into the blood. Ejection is removal of undigested food from the body as feces. Okay, so you all must be knowing all these terms and memorize it well, because this is commonly asked a lot. Okay. Okay, now moving on to the next one. What is physical digestion? Physical digestion is the breakdown of food. Oh my god, sorry. Okay, it's the breakdown of food into small pieces without any chemical changes to the food. There are no chemical changes to the food. It just breaks it down physically into smaller pieces. Why does it? So that there is increased surface area for enzymes to act upon. For example, let me give you an example. This is one box. If it is broken down to smaller similar boxes with the same volume, what is going to happen is that this surface area that is exposed, even this surface area is exposed, even this is exposed. So the surface area that is exposed increases. Here only it is this. Here it increases. Okay, the surface area that is exposed. I hope you all got my point. Okay, so it increases surface area of food for the action of enzymes, and the chemical digestion is basically going to happen faster. Ancestors, canines, premolars, and molars. Okay, this we will discuss right now. Discuss later. The diagrams we will do in the end for all the chapters. Okay, then. Okay, about the types of teeth, remember that ancestors and canines are used for biting and cutting, whereas molars and premolars are used for grinding the food. All right. Okay, we are going to do the diagrams later, but this I have to show you so that I can explain you. Basically, at the top part, this is the animal. Okay, the job of the animal is that it is the hardest substance which is made by the animals. Okay, and it is very hard to break it or chip it off, and it basically covers it. Okay, and it is very hard to break it off. But however, it can be dissolved. It basically bacteria comes on bacteria. If we basically eat sweet food, and if it is left on our tooth, the bacteria will come and feed on it, and it will basically produce acids which will dissolve this enzyme, this animal, sorry, and it will decay. Okay, then below the animal we have the dentite. All right, basically the dentite is not as hard as animal, but it contains cytoplasm and living cytoplasm. Okay, then next there are basically, let's talk about the cement. Okay, the cement, what it does is that it keeps the tooth embedded in the gum. Okay, this is the gum. Okay, so it keeps the tooth in place in the gum, and it has fibers growing out which basically attach the tooth to the jaw but not and allow it to move slightly when biting or chewing. Okay, then talking about the middle of the teeth, that is the nerves and the blood. Okay, where is it? The blood capillaries and yeah, the nerves. Basically, what they do is they supply the blood vessels, supply the cytoplasm in the dentite. We discussed dentite has cytoplasm, so it supplies them with the nutrients and the oxygen. All right, so yeah, this is all about the teeth. Okay, make sure you know the labeling. Okay, so before I start what all happens in our body in digestion, first you should be knowing that chemical digestion is a breakdown of large insoluble molecules into small molecules, and it takes place with the help of enzymes. Okay, it produces small molecules, small soluble molecules, so that they can be absorbed. Okay, now let's start with the whole digestion. I'm going to tell you it very quickly, very quickly. So basically, in the mouth, the teeth, lips, and the tongue, they play a role in digesting the food and biting it and grinding it, and it starts the physical digestion because it breaks the food into smaller pieces so that it can be acted upon by enzymes faster. Okay, then in the mouth also chemical digestion starts. Chemical digestion is making insoluble molecules to small soluble molecules, right? So that it can be absorbed, and basically takes place with the help of enzymes. And as I told you all this before, okay, that amylase breaks down starch to simple reducing sugar like glucose. Protease is break down protein to amino acids. Okay, this is something which I didn't tell you. Lipase breaks down fats and oils to fatty acids and cholesterol. So basically, in the breakdown of starch to amylase starts in the mouth, because the salivary glands makes saliva, and in the saliva there is a starch that is present, so it starts breaking it down. Then next it starts breaking down the starch to amylase, which is why if you keep the bread in long for your mouth and keep chewing it, so you can feel the sweetness of the bread because the starch is being broken down to glucose. Okay, now next, in the mouth, so physical digestion is also taking place and chemical digestion is also taking place. Now next, but remember, only chemical digestion of starch is taking place. That is, starch is being broken down to amylase, not any other, not proteases or lipase. That starts in the stomach. We will see about it. Okay, then next we have the esophagus. I'm going to tell you all very quickly. Okay, in the esophagus, okay, yeah, in the esophagus, basically peristalsis takes place, which pushes the food down to the stomach. Okay, and basically remember that there's a lumen in all of our alimentary canals, and even blood vessels, they have a lumen. Alright, so it's basically a space in the tube, middle of a tube of any body. Okay, the muscle relaxes to pass food into the stomach and then contracts to close it, which is basically the term given to this is called peristalsis. Okay, now esophagus is easy. First it goes to the mouth, now it passes through the esophagus. After the esophagus, okay, if you want, I'll show you guys a diagram. Just give me a minute. Okay, so since y'all were telling me to do it with the diagrams, so I thought why not showing you. By the way, this is the leaf one. We took a picture from Save My Exams, and this is the teeth. Okay, so this is the one. Okay, so in the mouth, this has extra labelings, by the way. You don't need to know so much. Anyways, you can refer the syllabus to know how much you need to know, you know. Okay, so it has the mouth. It starts with the chemical and the physical digestion. Okay, physical digestion because it's breaking it down to smaller pieces. Chemical digestion because starch is being broken down to amylase. Okay, then passes through the esophagus. Okay, which is close to the trachea. Then it goes into the stomach. Okay, so now what is happening in the stomach is very important, and you all must know that. Okay, in the stomach, it has basically strong muscular walls, which is going to contract and relax. Okay, I'm just going to contract and relax to mix the food with the enzymes and the mucus. Okay, it's going to contract and relax, and it contains all the enzymes. But one thing about the stomach is that it contains the hydrochloric acid. And as it is said in the syllabus, okay, if you see the syllabus, we'll come back to the syllabus. But for now, wait, I'll show it to you. Okay, just see it over here. Oh my God. Okay, basically all my drawings got erased, the highlights, it's fine. Okay, wait, see, this is the function of the hydrochloric acid. You should be knowing it. The hydrochloric acid in the gastric juice, it kills the harmful bacteria, microorganisms basically. Okay, so it kills harmful microorganisms and it provides an acidic pH for an optimum enzyme activity. Basically, there is this enzyme which in the stomach it is called pepsin. Okay, wait, I will tell you. Both. Okay, see, the proteases break down, as we saw here, the proteases break down protein to amino acid. But the protease which breaks down protein to amino acid in stomach is called pepsin. Okay, just remember it from Pepsi. And in the small intestine, it is called trypsin. So they're different enzymes that do the same job. This works in the stomach and the other one works in the small intestine. And the difference is that they require different pH. Pepsin works best in an acidic pH and trypsin works best in an alkaline pH. Okay. So the hydrochloric acid provides optimum pH for the pepsin in the stomach to work, and the trypsin becomes alkaline. We'll see how it becomes alkaline. Okay, for now, the function of hydrochloric acid, it has two functions: to kill the harmful microorganisms and to provide an acidic pH for optimum enzyme activity. The pepsin works the best at the pH of 2. Okay, now another role the stomach has, it churns the food, it mixes it really well. Now after 1 or 2 hours, now it would pass on to the duodenum. Okay, let's go back to the diagram. Okay, so see from the stomach. Oh my God. Okay, so from the stomach now it is going to go to the duodenum. Okay, this top part is called the duodenum. Please remember that. And the next one is called the ileum. Okay, this is the duodenum and this is the ileum. Please remember that the first one is duodenum and duodenum. The first one that is shown, and after that is ileum. Okay. Okay. You can just remember like D comes before I, so that's why. Okay, so the duodenum and the ileum together, they're called the small intestine. All right, so basically what happens in the duodenum, it is a part of the small intestine. Now we will be discussing about the small intestine. We have discussed about the stomach. The main role of the stomach is that it produces hydrochloric acid that kills the harmful microorganisms and it provides an optimum pH for the proteases enzyme to work. About the mouth, we discussed how it carries physical digestion and carries chemical digestion of starch to amylase, because the salivary glands produce saliva which contains the amylase. Okay, so next now in the small intestine is where, okay, basically the digestion of protein was started in the stomach. Pepsin broke down the protease broke down protein to amino acid. Now in the stomach, what is happening? That it is the part of the stomach, the small intestine. Okay, so what is going to happen in the small intestine is that there is a substance which is made by the liver and it is stored in the gallbladder. This substance is called bile. Okay, liver makes the bile. Remember, liver makes it and it is stored here. Okay, stored in the gallbladder. And when it is added to the mixture, when the food is passing from the stomach to the duodenum, basically this moves from the bile moves from the gallbladder to the duodenum in the bile duct. Okay, now it moves from the bile duct into the duodenum. So okay, now this bile, remember, this bile has a lot of role. Basically, this bile is alkaline. Okay, this bile is alkaline. This bile will cause the pH of a small intestine to become alkaline. And as we discussed before, trypsin works best in alkaline conditions. So the bile does the role of making the pH of the small intestine alkaline to allow trypsin, okay, to work best in the small intestine and to allow it to break down protein to amino acid and work at the optimum pH. Okay. Not only that, bile has more roles as well. Let's have a closer look at those. Okay. Okay, so what bile does is it is going to emulsify the fat. Okay, bile is made in the liver. It is going to emulsify the fat. What does it mean? It's going to break down large droplets of fat into smaller droplets. Okay, large droplets of fat to smaller droplets. Large droplet to smaller droplet. Why? So that it can be easily acted upon by lipase. Okay, so that it can easily be acted upon by the lipase. Now, where does this lipase come from? The lipase we discussed before is going to break down lipids, that is fats and oils, to fatty acids and glycerol, right? So where will it come from? Okay, so this bile is going, the enzymes that are going to break down the starch to amylase, okay, because starch, the breakdown of starch to amylase is done by the mouth. Okay, so I'm so sorry. The breakdown of starch to maltose by amylase is done by the mouth, right? By amylase is done by the mouth. But not all of it is broken down to maltose. And maltose, or you can also say glucose, by the way, it's not that big deal. You can just reducing sugar to be clear, okay? So it is broken down by amylase, but it's not completely broken down in the mouth. There is further digestion needed, chemical digestion needed, and that is done in the small intestine and even the digestion of lipids. So how is it going to do? The bile has played its role. Bile made it alkaline. 2 roles it has played. It made it alkaline and it emulsified the fat, that is, it broke down large droplets of fat into smaller droplets. Now what is going to happen? We need that smaller droplets of fat to be broken into fatty acids and glycerol by lipase, so that in that pancreas is going to come into role. The pancreas is going to make pancreatic juices, which will be added to the duodenum from here, and it's going to flow from the duodenum to the ileum. And in the ileum, what is going to happen is that all the nutrients will be absorbed into the blood. That is called absorption. But before that, the starch will be broken down to amylase. Pancreatic juices basically contains all the enzymes, okay? And this pancreatic juice flows along the pancreatic duct and into the ileum. This is a pancreatic duct, by the way, okay? This is a pancreatic duct into the duodenum it flows, all right? And basically it contains what do you say? It contains protease, okay, the trypsin it contains, okay, it contains lipase, and also contains amylase, which will act in the duodenum, and in the ileum, the absorption of the enzymes, of the nutrients will take place, all right? So now what is going to happen? Okay, so I forgot to tell you guys from where the pepsin comes into the stomach. The pepsin is created from the stomach walls in the liquid called gastric juices, okay? The stomach, the gastric juices is made by the stomach, and it contains the hydrochloric acid, and the hydrochloric acid has two roles: triggers the harmful microbes and also provide an acidic pH for pepsin to work at. And the gastric juices also contain pepsin, which is going to break down, of course, the proteins into amino acids, okay? Another thing that you should be knowing is that, basically starch is broken down to maltose, right? We discussed it. Maltose by amylase. Now, this maltose is further broken down to glucose by maltase. Okay, now where is this maltase coming from? This maltase is coming from the epithelium lining in the small intestine, is where this maltase is present, and it is going to produce glucose molecules, okay? And it is all just present in the lining, okay? It's just present in the lining and it stays over there, okay? It stays over there. It does not move from the surface. Maltose is digested on the surface only, okay? It's digested on the surface only, all right? It does not go into the lumen of the small intestine. It's just there on the surface of the epithelium. Okay, so that's all you should be knowing. Then trypsin, it works best in alkaline conditions, okay? And then fats and oils are insoluble in water, of course, so bile breaks it down. It emulsifies the fat so that it can be easily broken down, all right, by lipases, which will be acting in the duodenum. Okay, so these are the types of teeth: incisors, canines, premolars, and molars, okay? And this diagram, do we have it here? Do we have it here? No, we don't. Okay, wait, I'll show it to you guys. Okay, so see here, amylase breaks down starch to maltose, maltase breaks down maltose to glucose on the membranes of the epithelium lining. Okay, pepsin breaks down protein. Okay, we've already seen that. Bile is an alkaline mixture that neutralizes acidic mixture of the food and gastric juices entering the duodenum. Okay, providing a suitable pH for enzyme action. Small intestine is the region where nutrients are absorbed. Okay, it is basically the ileum. Okay, the ileum. All right, then the significance of villi, microvilli, of course, it increases surface area. The structure of villus, basically villus, it's easy. They're basically lacteals, and then there's arteries, and then there's veins, and the vein goes back to the hepatic. Through the hepatic portal vein, it goes back to the liver. Then most of the water is absorbed from the small intestine, and some is absorbed in the colon. Okay, we did not discuss about the large intestine. Basically, okay, I'll take you guys back to the diagram. Okay, it's pretty straightforward. The colon and rectum make the large intestine, and just remember, colon is before rectum, and then there's the anus through which the feces are excreted out of the body. But if you remember this thing I want you all to remember is that most of the water is absorbed in the small intestine. Many people say colon, which is a mistake. Just say it is most of the water in the small intestine, all right? Now, the transport of plants is a pretty straightforward chapter, to be honest. Xylem, transport of water and mineral ions, and support, and phloem is for the transport of sucrose and amino acids. All right, it's for the transport of sucrose and amino acids, and xylem is for the transport of water and minerals. Okay, then there are many structures in xylem that you should be familiar with. They have thick walls with lignin. They have lignin. They have no cell content. Okay, they have thick walls with lignin, which provides support. They have no cell content, so that water can flow easily, and their cells are joined end to end with no cross walls, so that it forms a long continuous tube. All right. Okay, some other features of the xylem are that it is wide, and so that carries large volume of water. There are dead cells. There are no end walls. There are broad space, strong cell wall, which helps it to withstand the high pressure. Okay, and lignin, of course, it prevents to collapse. Okay, so yeah, that's about the properties of about the structure of the xylem. So about the position. Okay, let's just see it. Wait. Okay, so you should be knowing the position of the xylem and phloem in a root. This is in a stem and this is in a root. Basically, the xylem is to the inside. Just remember, the xylem is always inside and phloem is always outside. So here xylem is inside and here phloem is outside. Remember this labeling. If we look at the... This is for a stem, okay? If you look for the stem, again, xylem is towards the inside and phloem is towards the outside. Wait. Yeah, xylem is towards the inside and phloem is towards the outside. So please memorize this and make sure you know how to label this, okay? And even in this, by the way, you should know in the vascular bundle, xylem is at the top and phloem is at the bottom. Okay, next thing is that the large surface area of root hair cells increase the uptake of water and mineral ions. Okay, they have a large surface area, of course. And then the way water moves is that it first moves into root hair cells, then to the root cortex cells, then to the xylem, and then to the mesophyll cells. Okay, the xylem carries it to the mesophyll cells. All right, now next... Okay, now transpiration is the loss of water vapor from the leaves. Water evaporates from the surface of the mesophyll cell, then into the air spaces, and then it diffuses out of the leaf through the stomata. Okay, water vapor loss is related to the internal surface area provided by the interconnecting air spaces between the mesophyll cells. So the greater the surface area, the greater the loss of vapor, water vapor loss, and the greater the size, the more the stomata, the more the water is lost. Now, basically, how is water lost? Basically, how is water lost from the leaf? Okay, how it moves upwards in the xylem. And basically, you should know that the transpiration, which is the lost water vapor from the leaf, creates a transpiration pull. Okay, it draws up a column of water and they are held together by forces of attraction between molecules, and xylem forms a continuous tube, and loss of water vapor from the leaf reduces water potential. So this is the mechanism. Okay, now increasing the temperature increases the rate of transpiration. Increasing the wind speed increases the rate of transpiration. Decreasing the humidity increases the rate of transpiration. How and why wilting occurs? Wilting occurs when the loss of water vapor from the leaf is greater than the uptake of water from the roots, which is when wilting occurs. We have discussed how the cells become flaccid and plasmolized. Okay, and yeah, this is investigations we will discuss in our paper 6 video. Okay, so remember all this. Okay, now moving to translocation. Translocation is the movement of sucrose and amino acids in the phloem from source to sink. Source is basically the parts of the plant that release sucrose amino acids, and sink is the parts of the plant that store the sucrose amino acids. Basically, for example, when a seed is developing, the store of the food will be in the seed, so that will be the source, and the developing parts will be the sink that which they're receiving. But when a leaf is fully develops and it has the leaves will be making the food, so they will be source, and the rest of the plants will be the sink. Okay, I hope you all get this. So different parts of the plant can act as source and sink at different times. Okay, sources are parts of the plant that release amino acids, okay, sucrose amino acids, and sinks are parts of the plants that store sucrose or amino acids. A translocation is the movement of sucrose and amino acids from the source to the sink in the phloem. Okay, so that was pretty quick compared to human nutrition. Okay, transport in animal circulatory system is a system of blood vessels with a pump and a valve to ensure one-way flow of blood. Basically, in single circulation of a fish, they have only a 2-chamber heart, okay? And double circulation has 4-chamber heart. In single circulation, the blood travels through the heart only once in a circuit of the body, and in mammals, it travels twice, so the high blood pressure can be maintained, okay? The pressure can be maintained, all right? Okay, another advantage is that it prevents mixing of oxygenated and deoxygenated blood, and it can flow at high pressure to the body. It's fast supply of oxygen, fast removal of carbon dioxide. Then blood is in single circulation. Basically, blood is at low pressure, and there's only one atrium and one ventricle, and there's no separation between oxygenated and deoxygenated blood. Okay, so this is about single circulation, and yeah. Okay, so basically, now this is the diagram of the heart. Okay, so you should be knowing that basically one diagram that really helps me remember is, okay, so see, these are the lungs. Okay, these are the lungs. Okay, and this is considered as basically, great, just give me a minute. Okay, so this is a diagram which was sent to me by one of my students. Okay, it is really helpful because for us, you can memorize it really easily. From the lungs, it comes through the pulmonary vein to the left atrium, then to the left ventricle, then through there to the rest of the body, back through the vena cava, and again back to the lungs through the pulmonary artery. Okay, the labeling of the heart I showed you guys before. Make sure you memorize it, all right? Okay, then another thing you should be knowing is that the left ventricle has a thicker muscular wall than the right ventricle. Okay, the left ventricle has a thicker wall than the right ventricle, because the reason is that because it pumps blood longer distance, like as you can see over here, it pumps it all the way to the body, with whereas right ventricle only pumps it to the lungs, which is closer to the heart. And then remember what are atrioventricular valves. They're present between this atrium and the ventricle. This valve is called atrioventricular, even this is called atrioventricular valve, and you don't need to know the names like bicuspid and tricuspid, but just know that this is atrioventricular valve, and this one the semilunar valves are present between the open entrances to the aorta and to the pulmonary artery. So it's present basically here and it's present here. Those are called the semilunar valves. Okay, valves. And now moving on next. Okay, let's get back to our syllabus. Okay, now septum is very important because it separates the oxygenated and the deoxygenated blood. Functioning of hearts in the terms of contraction of the muscles of the atria. Okay, when the atrium muscles contract, the ventricles relax and all. Okay, and the blood is pumped away from the heart in the veins and returns in the vein. It's pumped away from the heart in the arteries and it returns to the heart in the veins. Activity of the heart may be monitored in listening to the ECG, listening to the valves opening and closing, and even to the pulse rate. Okay, so these are three ways in which the activity of the heart can be monitored: ECG, pulse rate, listening to the sounds of the valve closing. Make sure you know all three of these. Okay. And yeah, septum is important, separating oxygenated and deoxygenated blood, pumped away from the heart and arteries and returns to the heart in the vein. Okay. Now, coronary heart disease is basically a blockage in the coronary arteries. And what are coronary arteries? Coronary arteries supply the heart with the oxygen and with the oxygen because it needs with oxygen, okay, and nutrients and oxygen that the muscle needs. Okay, so it needs a constant supply of these so that it can undergo aerobic respiration. Because the muscle is so thick, so it would not be able to diffuse to all them quickly enough. Okay, so yeah, that's why. Okay, and the possible risk factors, sometimes, you know, what happens is that the coronary heart artery, which supplies the heart with the nutrients and oxygen, it becomes blocked with fat deposits. So then what, that is very dangerous and it can lead to a heart attack, okay, and even death sometimes. I mean, yeah, most of the time. No, I don't know. Okay, so what are the risk factors that are associated with this? The risk factors that are associated with this is diet, lack of exercise, stress, smoking, gender predisposition, age, and sex. Basically, genetic predisposition. Basically, diet. Your diet matters a lot. If you eat a lot of saturated food with a lot of fat, okay, it can affect your arteries. So eat good diet, eat good fruits and vegetables. Okay, everything in moderation is the best. Lack of exercise, stress, smoking, and then genetic predisposition. Like if your ancestors have it, you have a lot of high chance to have it. And of course, age, as you grow, you have more chances of getting it. And as sex, of course, men are more likely to have it than women. Okay, then diet and exercise play a diet and exercise play a very big role in controlling and reducing the risk of this disease. Why? Because it reduces the stress, okay, lowers stress, okay, and then it lowers the BP, the blood pressure, okay, and then what it also does is it heart muscles, okay, muscles become stronger. Okay, so these are the three reasons why exercise is good for you and you should control your diet and eat more fruits and vegetables. So basically, diet and exercise, they lower your blood pressure, they lower the stress, and what they do to your heart muscles is that it makes your heart muscles stronger. Then effect of physical activity on the heart rate, it increases the heart rate basically. And yeah, the three ways in which to measure ECG pulse rate, listening to sounds of the valves closing. Okay, so now talking about the blood is made up of blood plasma, okay? The blood is made up of plasma and it is also made up of cells, okay? So basically these cells, the plasma makes most of the part and their cells, and there are three types of cells. Basically, in cells you have red blood cells, okay, RBC, and white blood cells, and you have platelets, and white blood cells are further divided into, phagocytes and lymphocytes, okay? Phagocytes. And you also have lymphocytes. Okay, so let's have a closer look at what this all is. So basically, when we are talking about these are blood vessels. Okay, let's have a look at the blood first. Okay, basically blood is made up of red blood cells, white blood cells, platelets, and plasma. Okay, remember this. Okay, identify red blood cell, white blood cell diagram, we'll see that later. Red blood cells is used in transporting oxygen, and there is a role of hemoglobin because hemoglobin combines with oxygen to form oxyhemoglobin. White blood cell used in phagocytes and phagocytosis and antibody production. Platelets are used in blood clotting. Okay, plasma is used in the transport of blood cells, ions, nutrients, urea, hormones, and carbon dioxide. Remember this: blood cells, ions, nutrients, urea, and carbon dioxide. Okay, then lymphocytes, basically they produce antibodies, and phagocytes, they directly engulf the pathogen. Okay, they directly basically eat the pathogen, whereas lymphocytes, they produce antibodies, and which will basically, what they will do is they will make the pathogen stay in the place so that it can be engulfed by the phagocyte. Okay, so basically white blood cells, they protect our body and they prevent us from getting diseases, and they do this by two processes, by phagocytosis or by antibody production. If it's phagocytosis, then phagocytes are involved, and if it's antibody production, the lymphocytes involved. What phagocytes do is they just eat the pathogen, like engulf it, and that is called phagocytosis. Okay, now what do platelets do? Basically, it says here details are not required, but for extended you need to know the details. Okay, so what happens is that the platelets are helping clotting. Okay, what is blood clotting? Basically, platelets are involved, okay, and what they do is that they convert the fibrogen to fibrin. Okay, so the fibrogen is soluble, so they convert it into insoluble, and what it is going to do, insoluble, what it is going to do is that fibrin will form a mesh. Okay, this insoluble fibrin will form a mesh and will wrap the all the blood cells, and that is going to heal, and it is going to form a scab, and it is going to heal the cut. So that's all about platelets. Now, the role of blood clotting is that it prevents the entry of pathogen and prevents loss of blood. Okay, so pathogens cannot enter our body and cause harm to us because they're always present around us and we don't even know about it most of the time. So that's why blood clotting is basically going to stop the pathogen from entering our blood, and it is also going to prevent the yeah, and it's also going to prevent the loss of blood. Okay, and the clotting we did. Okay, we look at the diagrams later. Okay, so we'll have a look at the diagrams right now. Let's have a look at it. Okay, it's pretty straightforward. This is, I hope we have those. Yeah, we have it. Okay, so this is the red blood cell. It basically has no nucleus. All right, I forgot to mention that. And it basically has no nucleus. The lack of nucleus is so that there's more space for hemoglobin to carry oxygen, and it is a diconcave disc shaped. Okay, and it has a large surface area. This size gives them a large surface area, and the hemoglobin is a protein, right? We already discussed that, and it contains iron. And of course, we already discussed also that if we have less iron, we'll have less hemoglobin and we will get anemia. So make sure you have more hemoglobin by eating liver, red meat, egg yolk, dark vegetables, if you all remember. Okay, now this the red blood cell is disc shaped. It has lack of nucleus, so that can carry more hemoglobin. Okay, now looking at the white blood cell, this is the lymphocyte. This is the phagocyte. I'm so sorry. This is the phagocyte. This is the shape of the phagocyte. Is a lobed structure, and the shape of, yeah, you can see the difference between them. There's no picture of lymphocyte. That's crazy. Okay, I will show you the difference between phagocyte and lymphocyte later. Lymphocyte is basically like a normal looking cell, but when you look at phagocyte is a three-lobed structure, and it engulfs the pathogen. You can kind of get it from its shape that it engulfs the pathogen, you know? It looks like a mouth, you know, to me. Like, I don't know if you can imagine it. Anyways, okay, so now the platelets, they're small fragments with no nucleus, okay? These platelets, they have no nucleus. They're just small fragments, and they can heal the cut, and they're basically used in blood clotting. And again, this is a white blood cell, red blood cell, plasma, platelets, okay? And yeah, so you should be able to see it even if it's not colored, because Cambridge loves to give that and annoy you, of course. Pisses me off, you know, when they give like black images. Not being a racist, I mean, like, you know, it's difficult to see. I hope you get my point. Okay, so now talking about this, what do you say? Talking about arteries and veins. Okay, there's a lot, there's not much actually. You just need to know it. Basically, arteries, okay, I'll be telling you. Arteries, they have a thick muscular wall. Okay, basically, first of all, they carry blood away from the heart, the arteries, okay, like the aorta, pulmonary artery. Okay, they're carried away from the heart, and they have thick walls, okay? They have thick muscular walls to withstand high pressure, so that they can withstand the high pressure, because especially aorta, it has to carry to all the parts of the body. Then they have elastic tissue, okay? They have elastic tissue which can stretch and recoil, okay? Which can stretch and recoil to allow the, what do you say? Yeah, it has elastic tissue to stretch and recoil. Narrow lumen, okay? Narrow lumen, it has a narrow lumen so that it can carry the blood at high pressure. Okay, so it has thick walls to withstand high pressure, elastic tissue that can stretch and recoil, and it has narrow lumen. And if you look at the veins basically, they have thin walls, okay? They have a wide lumen first. Let's talk about wide lumen. Wide lumen so that there is less resistance to blood flow and can carry large volume of water. And also you can say large lumen, okay? Large lumen. Then you should know that they have valves. And by the way, they have a thinner muscular wall compared to arteries, okay? And they have valves to prevent backflow of blood, because the blood flows way slower in the veins than in the arteries, so it can backflow. So to prevent backflow, they have valves, okay? So they have valves, and they have a large lumen to carry a large volume of water and prevent the resistance to the blood flow. Arteries, they have thick muscular walls that can stretch and recoil, and They have elastic tissue actually that can stretch when you cut, and they have thick muscular walls to withstand the high pressure, and they have a narrow lumen so they can carry the blood at high pressure. Okay, now moving on to, this was about arteries and veins. Now moving on, what the hell? Okay, now moving on to the capillaries. Capillaries, basically they're just one cell thick, okay, to allow oxygen and carbon dioxide and all the gas exchange, you know, to take place in the capillary. Okay, so we have now discussed the function of capillaries. Okay, vena cava, aorta, pulmonary artery, and this diagram, you know, it is this all is in the diagram. The diagram PDF, I'll attach it in the description. Make sure to go through it. Okay, even this one. Okay, so the function, structure of capillaries related to its function, of course, because it has thin walls, so that it can easily, you know, it's only one cell thick actually, so that gas exchange can take place easily and greater rate of diffusion. Okay, now blood is also done. Now moving on to diseases and immunity. This is also pretty easy chapter. Pathogen is a disease causing, what the hell? Okay, pathogen, what the hell? Okay, pathogen is a disease-causing organism. Transmissible disease is a disease in which the pathogen can be passed from the host to another, like from one person to another. And there are ways in which the pathogen can be passed by direct contact, like through blood and body fluids. And then there's indirect contact through air, animals, okay, and contaminated surfaces and food. Like if someone sneezes into the air and you breathe it in, then you can get what? A disease. Tank. Okay, then there are body defenses. Okay, you have skin, you have hair in the nose, you have mucus, you have stomach acid, and you have white blood cells. Okay, so we saw that stomach acid contains hydrochloric acid that is going to kill all the acid, and it is going to lower the pH. Okay, then moreover, what does the skin do? The skin prevents it, acts as a barrier. Mucus traps the bacteria. Hair in the nose also trap the bacteria. White blood cells in the body defense, you know that it kills the pathogen. You know what white blood cells do? They detect the foreigner as the pathogen as a foreigner, and they just attack it. All right, but it takes time for the white blood cells to make antibodies. Okay, we'll have a look at it right now. Okay, by direct contact through blood and body fluids. Okay, and by indirect contact, we just discussed contaminated surfaces, food, and animals. Okay. Okay, so now as I told you, when a pathogen enters the body, okay, the white blood cells will identify it as foreigner, okay, and it is going to make something that is called antibodies. Okay, we discussed about it before, but I did not tell you what are antibodies. Okay, antibodies. Okay, antibodies are basically, let's have a look at the definition. They are proteins that bind to the antigens for direct destruction of pathogen or marking of pathogen for destruction by phagocytes. Okay, the antigens are basically present on the pathogen. Like if I draw pathogen, this, like this, okay, this is looking how coronavirus looked, something like this. Okay, so this thing, okay, these are called the antigens. Okay, so what the antibody is going to do, antibody is going to look something like this. Okay, and it's going to engulf this okay, so it should actually be complementary to the shape of the antigen. Okay, so it's going to look like this, and it's going to engulf this antigen, and it's going to directly eat, it's going to what is it it's going to do? It's going to lead to direct destruction of this pathogen, or it is going to mark this pathogen for destruction by phagocytes, so that phagocytes with the lobe nucleus can basically come and eat it. I don't have to draw the lobe nucleus. Okay, so then the phagocytes can eat this. Okay, and of course each pathogen has its own antigens which have a specific shape. Now there are two types of ways the body can defend us. There's active immunity, okay, by antibody production in the body. When antibodies are made, basically, okay, so basically when the lymphocyte, when the anti-white blood cells detect a pathogen and when they make antibodies, something called memory cells are produced. Okay, these memory cells, they remember the pathogen forever, and whenever you get the disease again, they will fight against it. Okay, but that only happens in active immunity, and you can get active immunity by specific antibodies. Okay, they have complementary shape, they fit specific antigen. Okay, you can get a antibody by the active immunity by infection by pathogen or by vaccination. When you get infected by a pathogen, and if you survive that pathogen, hopefully the antibodies are made now and memory cells are produced. Okay, whenever memory cells are produced, it's active immunity. Okay, so now memory cells are produced, and now you are gonna be immune to that pathogen. If you ever get that pathogen again without you getting sick or getting any fever, the antibody, the white blood cells will fight that pathogen. But and also by vaccination. Why? Because in vaccination, a weakened pathogen or its antigens are put into the body, and this will stimulate an immune response. And the same way, it looks like you actually got the disease, but it's a weakened pathogen which can barely cause any harm to you. So it's just so that memory cells are produced and it can help you, and it's of course going to give a long-term immunity. Okay, and there's a lot the role of vaccination in controlling a disease, the controlling the spread of disease is very important. Why? Because if people are vaccinated to a disease, the pathogen will have very few places to live and grow, and therefore the spread of disease will be decreased. Okay, so non-vaccinated individuals will also get protection as a result of herd immunity, because the pathogen will have less places to go to. Now, passive immunity is opposite of active immunity, and it's a short-term defense against a pathogen. And you get the antibodies from another individual. Your body does not make the antibodies. So when antibodies are not made, there is no immune response triggered. So memory cells are not produced in this. When memory cells are not produced, you will not get a long-term immunity. If you again get that disease, if you get that disease, God forbid, your body will have to fight against it, and you will get sick, because that time the body will take time to make antibodies. Okay, and there are many chances, if it's like a dangerous disease, the disease might already cause harm before the antibodies are made. A lot of harm before the antibodies are made. That is why we all got vaccinations for COVID-19, and it was a weakened pathogen, so that if God forbid we get the original strong pathogen, it enters our body, we can easily fight it because memory cells are already produced. Alright, so it's a short-term defense against a pathogen, passive immunity, because you just get antibodies from another individual, and it's a short-term, it's not going to last long. You get it from your mother across the placenta and in breast milk. Okay, so it basically helps the infants. Its importance is that it basically helps the infants in the early age, because they can't make antibodies that time. Okay, and it basically, it provides all the nutrients to the baby, and it provides them with passive immunity from an infection, of course, and before the baby can start making its own. And memory cells are not produced in passive immunity. Of course, everybody should know that, and they're produced in the active immunity. Okay, we didn't do that. The importance of controlling the spread of disease: you should have a clean water supply. Nowadays, watery, clean water, and if the water supply is not clean, it can cause cholera. We'll look at it right now. And there should be hygienic food preparation, good personal hygiene, waste disposal, and sewage treatment. I don't think there's something that needs to be explained here, because yeah, it's simple. You should have good personal hygiene, and you should keep yourself clean, because there's oil secreted by your body which can cause the pathogens to come to your body. Okay, cholera is a disease which is caused by a bacterium which is transmitted in contaminated water. Okay, this bacteria is present in contaminated water. Okay, and this cholera bacteria, it basically produces a toxin that causes the movement of chloride ions into the lumen of the small intestine. Okay, they move into the small intestine, and is going to cause water to move. What is going to happen when chloride ions increase in the small intestine? Okay, this is the small intestine. What is going to happen is that the water potential will decrease here. Okay, because the solute increases, the water potential will decrease here, and the water potential will basically be more here. So water will move from to the small intestine, which will cause a lot of water loss from the body. Okay, it will be lost to the large intestine. Okay, and it will cause diarrhea, dehydration, loss of ions from the blood. You need oral rehydration therapy for this, and if you don't get it ASAP, as soon as possible, it can even lead to death. Okay, so that's all about diseases and immunity.